Hello, everyone. Welcome to the CRB1-Associated Inherited Retinal Diseases webinar. We're going to get started in just a moment. For those of you who are coming on, we have a UniRare flyer up on the screen. We'll be talking about it a bit later. And if you would like a copy, please feel free to email and we will send you a copy. We'll get started in just a moment here. Okay, hello everyone, and welcome to our CRB1-Associated Inherited Retinal Diseases Forum. My name is Amanda, and just to kick off the presentation, we will have just a few logistical details. So all of the participant lines are muted, no video for participants, so don't worry. Our speakers will have their videos live, but they will explain anything that is on their screens, slides, presentations, all of that. The control bar is at the bottom of the Zoom interface. We are recording currently, and captions are available at the bottom of the Zoom, and at the bottom right, there is an ellipses that's labeled More. When you click on that, you're able to access captions. So you can change the language on your captions and you can turn them on or off. So then also on this bar, there's the Q&A function. We'll address those questions towards the end of the presentation, but throughout you can submit your questions. And we will mention that again later on. To prevent those controls from hiding, you can press the Alt key on a Windows keyboard, or you can press the Command and backslash simultaneously for the Mac keyboard. Okay, so to get this presentation started, I would love to introduce Tabby Mitchell. Tabby?
Aloha, can you guys hear me okay? Yeah, okay. Welcome, everybody. I'm coming to you guys from the middle of the Pacific on one of the islands of Hawaii. We're on Oahu. It's 7 a.m. here, and I know that we are crossing a lot of time zones today, and for that, we're all excited. In Hawaii, when we start something like this, we would say, Ikomo mai CRB1 ohana, and that translates to welcome. That's Ikomo mai, and we all know ohana, right? Ohana means family. And family means no one is left behind or forgotten. And I think that's really kind of cool to kind of open us today, simply because that's what this forum is all about. That's what we hope that we can begin to do, is to all come together and to share information and unite and move together as one great big CRB1 ohana. I know a lot of you guys from Foundation for Retinal Research, whenever we would have all of the LCA family conferences and all of that effort with David and Betsy. And I remember very clearly, almost like yesterday, when we all got to hear live from the stage, Dr. Bennett and Dr. Acquire, when they were talking about Lancelot and how they had cured blindness in the dogs. And we actually got to pet Lancelot. We got to see Lancelot, all of that, very exciting times. And that excitement has carried us through to where we are today. Hopefully you guys know about the LCA Family Conference coming up in Indianapolis in the States. I believe that's the end of June 23rd, 24th. I know you can sign up on Hope and Focus. And it's kind of funny because that takes me back to where I was whenever we received my diagnosis for our daughter. I actually have 5 kids. And I used to work in Indianapolis, believe it or not. Eli Lilly is based there, and that is a pharmaceutical manufacturing research and development hub for Eli Lilly. It's actually where the only place where they had operations in the United States for a really long time. And that was my background. That's what I was doing whenever we gave birth to I gave birth to our 5th daughter. And she is the one that was diagnosed with LCA. And she is that birth and that situation, that culminated basically with me leaving my career in Eli Lilly. And I laugh and joke all the time that we have such a big family. We have 5 kids. And we did that because we had such nice benefits with Eli Lilly, and they had great maternity benefits. And they also had the opportunity for employees to take a 3-year leave of absence. I don't know if this is still the case. I haven't touched base with them lately, but I never viewed myself as that employee that would ever take that benefit. But whenever I gave birth to Emmy, she had a really large bulbous ear tag on one ear, and she failed her hearing test when we were in the hospital. She was a C-section, so we didn't get to leave early. We were there for like 5 days. And then we began to notice that she had vibrating eyes. As many of you have experienced, she had nystagmus. And so one thing led to another, and I ended up taking advantage of that 3-year leave of absence. And what I did during that 3 years was basically the classic diagnostic odyssey in rare disease. And we tracked down and identified that Emmy had LCA and that she was a CRB1 mutant like the rest of us. And with that, one thing led to another. And, we're all living the dream. We're in that club that none of us wanted to be in. We're learning how to wrangle blindness. We're learning how to change the world with all of the people that we interact with for our kids and for ourselves as adults, and to raise those expectations of blindness. And we're all living that side of it. But the other side of that fence is the research side. And the other side of that fence is the hope. And the hope comes in with going back to Lancelot, right? And the dogs, and going back to what can we do and how do we move that needle? And that's why we're here today, because we need each other. Really, we need each other to come together. We want to be able to share all of the innovative things that Foundation Fighting Blindness has been doing over the years and that we're currently doing. And we want to bring everybody into the fold so that everybody can understand where that research is and where that research is going and what it's going to take to continue to move that research forward, because it is indeed a very exciting time for vision research. I know I field lots of calls from lots of parents all the time going, hey, when's the clinical trial? When can I sign up? And it's just not that simple. And yes, we are obviously trudging forward with that, but it takes a lot of funding. It takes a lot of effort. And it's really exciting that we'll have Dr. Winholtz with us later today to go through some of his research. Oddly enough, the last time that I interacted with Dr. Winholtz was here on Oahu when I think it was the ARVO conference. So it's good for all of us to be able to get back and do all of the things. We've all survived Corona. We're moving right along. And then one of the other things that we've been doing over the course of time that Emmy was kind of a trigger for was us trying to find a non-traditional way to be able to fund vision research. And what we ended up doing was kind of a tag team, kind of like Dr. Bennett and Dr. Aguirre that I mentioned earlier. We did a husband and wife tag team. And my husband and I started a tech company. And the whole reason that we did it was to be able to make a profit to fund vision research. And over time, we also fund programs that support blind kids because there's such a need for that. And I'm really excited to be able to say today that later on in this webinar, you're going to hear from specific, I don't know exactly who's going to deliver the information or how the program is going to work, but our company is prepared to match any donations that we can raise to help move this research forward for CRB1. And we're going to match up to $100,000. So I'm hoping that you guys can heed that and get excited by that. I have 28 people that work in our company as cyber and IT people. They don't necessarily have anything to do with vision research or Emmy's situation, but they take it to heart and they know exactly why our company exists and they know why we do what we do. If any of you guys are out there and you're looking for positions, I urge you to go check our website, because we're always hiring. We're always trying to find a way to bring that money forward so that we can take that money into vision research. And so with that, one of the big things over the course of time that I would do when I was working with Steve Rose, who was our past scientific, I don't know his title, past scientific director or whatever with FFB, he knew that my desire and dream was to be able to have somebody that could focus within FFB and to focus on CRB1 research and to focus on what's being done, what needs to be done, what are the missing holes, what are the gaps, how do we tie it all together? Those of us that have blind kids, it's much like a case manager, right? We manage their case, we manage their IP, we figure out what training they need. I wanted that for CRB1, and that was a personal dream that I had. And I'm very excited that in our last in our last quarterly meeting that we had with the foundation, and we were talking about research, we introduced Angela Bowman. So Angela has now onboarded with Foundation Fighting Blindness. It is one of her tasks and one of her roles within the organization to do just that, to manage what's going on in CRB1. And with that, I'm going to turn it over to Angela. Again, ikomo mai, everybody.
Great. Thank you so much, Tabby, and thank you for sharing your story with us and for your generous offer of a match to any funds that are raised today. As Tabby said, I'm Angela Bowman. I'm the Science Advocacy Lead. That's my official title at the foundation. And yes, CRB1 is one of the genes that I'm tasked with managing. Thank you so much, everyone, for being here with us today. Just a quick note to those on the call, I won't be using slides, so you don't need to worry about following those. I'm excited too that we have Dr. Jan Winholz with us today, but before I turn things over to him, I wanted to share an update on some of the activities at the foundation around CRB1, as well as research on CRB1 that was presented at a recent Vision Research Conference. Those of you who were on the last forum call in November may remember that I joined about 9 months ago. And I have been diving into the CRB1 research since then, and I'm working on a landscape analysis document for CRB1 that I plan to have complete by the end of June. And this document, which will be shared with the community, both scientific and constituent patient-wise, will really help us identify gaps in research and next steps. You may also remember that the foundation funded the analysis of the CRB1 gene and protein expression in pigs to determine if they would be a good animal model to develop for CRB1-associated disease. Unfortunately, the CRB1 mouse models don't fully recapitulate the phenotype that we see in humans, so we need better models. Those studies, which were carried out by Dr. Marie McCall at the University of Lexington, found that the CRB1 expression patterns in pig and humans are very similar, indicating that the pig would be a good model for further development. So currently, the National Swine Resource and Research Center has begun designing the tools needed to make a pig carrying a CRB1 mutation. And we should know by this summer if their strategy has been successful. And the foundation has already committed funds to Dr. McCall to characterize those mutant pigs to understand their visual phenotypes. As Amanda mentioned, there was a flyer on the screen when you logged on about the UniRare Natural History Study. This is a natural history study that the foundation has just launched that will follow 1,500 patients with rare inherited retinal dystrophies. The goal is to get a snapshot of many different retinal diseases, and patients with CRB1 mutations are encouraged to participate so that we can understand CRB1 disease progression and patient variability. If you have any other questions about the UniRare study, we can mail you a copy of the flyer that's on the screen, or you can find more information on our website, or you can always email me directly and I can get you the information you need. As I mentioned, I was recently at a large conference for vision researchers at the end of April. There were close to 10,000 vision researchers there, and there were a number of studies focused on CRB1 that were presented at the conference. I just want to highlight a few of them, but I was thrilled to see that there were so many different researchers working on CRB1. As I mentioned before, we don't have good animal models to help us understand CRB1-associated disease. We also often can't tell immediately how a patient's mutations are affecting their CRB1 in their eyes, how those mutations are causing disease. So researchers from the Lions Eye Institute in Australia developed a platform that allows them to study CRB1 expression and function in patient skin cells in the lab. In a pilot study with their platform, they examined 11 different patient samples and were able to show how the different mutations change CRB1 expression. And they hope that this platform could be used for all CRB1 mutations to better understand them. I would next like to highlight two studies from Dr. Peter Quinn from Columbia University. Dr. Quinn was a graduate student with Dr. Winholz and was a postdoctoral fellow with Dr. Stephen Sang, whom the FFB has supported extensively. Dr. Quinn presented two posters describing therapeutic strategies for treating CRB1-associated disease. The first study explored if specific mutations in CRB1 patient cell lines can be corrected with prime editing. Prime editing is a modified form of CRISPR genome editing that allows specific mutations to be directly corrected in a patient's DNA. Dr. Quinn's team developed induced pluripotent stem cell lines from patients and prime editing tools for 2 of the top 10 most common CRB1 mutations. Those were the cysteine 948 tyrosine mutation, which is the most commonly reported mutation, and then the glycine 1103 arginine mutation. And they found that they were able to correct the CRB1 mutations in these cells, though not with 100% efficiency. They are currently working on developing a strategy that will allow them to deliver these tools directly to the eye using 2 adeno-associated viruses. One caveat to this study is the fact that every drug targets a different mutation, so every mutation would need a different drug, and this is challenging to think about from a time and financial perspective. However, this does lay the foundations for using prime editing in patients. Dr. Quinn's second study explored a gene therapy approach. You may know or remember that there are different flavors of CRB1 expressed in the eye called isoforms. And the team first looked at which cells in the human eye express which isoforms. They found that CRB1A is largely localized to Mueller glia cells, and CRB1B is mostly found in photoreceptor cells in the adult retina. This is important because it might provide clues as to which cells need to be treated with which CRB1 isoforms. Importantly, they also found that when they used gene therapy to deliver CRB1A and CRB1B together, they saw improved retinal function in a CRB1 mouse model. And this project was recently awarded a $1.25 million grant from the National Eye Institute. And we're looking forward to hearing updates on this exciting project. So I would now like to introduce our scientific speaker, Dr. Jan Winholz. I'm sure that name is familiar to many of you in the CRB1 community, as he has dedicated most of his career to studying CRB1. Dr. Winholz received his doctoral degree from the University of Groningen in the Netherlands. And then after completing 2 postdoctoral fellowships, Dr. Winholz began his independent lab at the Netherlands Institute for Neuroscience in the Netherlands Ophthalmic Research Institute in 2000. In 2015, Dr. Winholz moved his lab to Leiden University Medical Center, where he is currently a group leader. Dr. Winholz will also be joined by one of his clinical colleagues, Jessica Karuntu, who will speak about CRB1 natural history studies ongoing there. The foundation has provided support for Dr. Winholz CRB1 research over the years, and we are so pleased to have him here today to share his recent work. I believe that Dr. Winholz may be using slides, but his talk can be completely understood without being able to see his slides. Dr. Winholz?
Yes, thank you for the very kind introduction. I will try to give this presentation without showing the slides. So if that's fine with the organization, so I will do it without the slides. So what we've been doing in the past years is studying the action by another family member called CB2 to overcome the loss of CB1. And we've done studies in retinal organoids, so iPS-derived retinal organoids from human patients. And these patients were retinitis pigmentosa patients. But first, let me inform about a disclosure. We have a patent on the use of the CB2 gene for gene therapy purposes. And as already was told likely many times to all of you, the CB1 is a relatively large gene. It is placed in Mueller glial cells and in photoreceptor cells. The total size of the protein is 1,406 amino acids. This is quite a lot of building stones for a protein. But this protein, whereas there's a dogma of that it would not fit into the AAV, it does fit in the AAV. We've shown already in a patent in 2013, and also in a publication in 2014, and a publication in 2015, and a publication in 2021, and now a very recent publication that this gene does fit into an AAV vector. It needs a shorter promoter, a shorter CMV promoter, it is that we call it. It can also be expressed with another short promoter, but so there should be no dogma about that this gene would not fit into an AAV. So as well, the CRB1 gene, as also the CRB2 gene, they both fit into AAV vectors. So in the family of the CRBs, there is CRB1, which is 1,406 amino acids, and there's CRB2 that is 1,285 amino acids. So the size is a little bit shorter of the CRB2. It can be expressed using a so-called full-length CMV promoter in nuclear cells and in photoreceptor cells. And there's also a third family member that is called CRB3A. This is a very short one that lacks the typical very large extracellular domain that is present in both CRB1 and also CRB2. So these two proteins, CRB1 and CRB2, on protein level, they are very, very similar. Especially at the C terminus, they are like two twins. They are very similar. We also looked at the structure of the CRB1 and the CRB2, and the CRB1 also has a shorter form that lacks a few regions in the CRB1 gene, and this particular shorter form looks almost exactly the same as the CRB2. So the CRB2 looks very similar to the CRB1, and it also has very similar functions, as I will show, as I will discuss soon. So in the past, we've done many studies on mice, and we've generated mice that lack the CRB1, so we could nicely see in the control mice the CRB1 protein, whereas just the CRB1 protein was lost in the CRB1 knockout mouse. And these mice develop a very particular phenotype of rows of photoreceptors that just are not localized anymore. So the nuclei of the photoreceptors, they are not localized anymore in the nuclear layer. They slip out into the subretinal layer. That is a very typical phenotype for the CRB1 knockout mouse. In the past, we've also generated CRB2 conditional knockout mice, and also these conditional knockout mice, so compared to the wild-type mice, they show very nice localization of the CRB2 at the location where also the CRB1 is localized, whereas just in the conditional knockout, this protein is gone. And in these conditional CRB2 knockout mice, we see a phenotype that is very similar to the phenotype observed in the CRB1. So where are these proteins localized? So, and there was a problem at a certain time that just in the mice, we found a different localization than in the human. In the mouse, the CRB1 is only localized in molecular cells, whereas the CRB2 is localized in molecular cells and in photoreceptor cells. And what they do is they mediate also some adhesion between the molecular cells and the photoreceptor cells, also adhesion amongst the rod photoreceptor cells, and adhesion between rods and cones, and adhesion between myoglial cells. So that is part of the function that these CB proteins have. That's very important from all of us. So we have about 12 different mouse models, all kind of variants. And what our studies showed is that one needs a particular threshold level to have normal retinas, versus the levels of the CB or the CB2 proteins are not sufficient. Then first, there is a phenotype very similar to retinitis pigmentosa, versus when the levels of CB1 and or CB2 are very low, then we see a typical Leber congenital amaurosis phenotype. So in LCA, the levels of the CB proteins or of functional CB proteins is very low. Whereas in retinitis pigmentosa, there are still CB proteins, but likely not sufficient. So in the human, the localization of these proteins is different. We've done studies on adult non-human primates, and we observed that the CB1 and the CB2 are both present in myoglial cells and photoreceptors. So this is also different from what I just heard from what Pete Quinn reported. So he himself did studies also, or he was involved in these studies when he was in my lab. So we determined that the CB1 and CB2 proteins in the adult human primate are present in both cell types. And also in the fetal human retinas and in human iPS-derived retinal organoids, we see that the CB1 and CB2 proteins are present in myoglial cells and photoreceptors. From our very recent studies, wherein we did gene profiling of CB proteins in the iPS-derived retinal organoids, we do see that the levels of CB1 and CB2 are about the same in photoreceptor cells and in myoglial cells, but the levels of CB2 are lower in myoglial cells. They're certainly there, so there is CB2 in myoglial cells, but the levels of CB2 are much higher. Well, so we obtained in the past proof of concept in 3 different CB mouse models. So these we published in 2015. The title of that paper was Gene Therapy into Photoreceptors and Myoglial Cells Restores Retinal Structure and Function in CB1 Retinitis Pigmentosa mouse models. And the other paper was published in 2021, also a paper on a mouse CB model. AV-CB2 protects against vision loss and inducible CB1 retinitis pigmentosa model. So, but what about the human disease model? But first, let me tell, so what is this about? Why the CB2 and why not the CB1? Well, I told you that both of these genes do fit in AV vectors. And both proteins can be efficiently expressed upon infection of these AV vectors onto the retina. The CB1, for the CB1, we use a minimal CMV promoter or a small CRK1 promoter. For the CB2, we use a full-length CMV promoter. And we even found that the minimal CMV promoter was even stronger than the full-length promoter. So we certainly obtained good levels of expression of the CB1 in the retina. So why then using the CB2 instead of the AV CB1? In 4 of our publications, we described retinal toxicity in vivo when overexpressing the CB1. So we described that in the patent application in 2013. We described that also in publications of proof of concept in 2015 and 21, and in a paper in 2014. So for us, the CB1 is a no-go because of toxicity. So it's not because it does not fit into the AV, it just is, in our view, not safe to use. And we did not observe any toxicity when overexpressing CB2, but we obtained clear protection when using CB2 in the CB1 retina. So the AAV CB2 outperforms the AAV CB1. So the CB2 is the best gene to use. So I will next discuss that CB2 protects in cultured human CB1 retinitis pigmentosa model retinal organoids. So that's what I will discuss today. So CB2 and also CB1 used in a cultured model, both can rescue the retinal phenotype. What I will not show today, but we have the data and we will submit that paper within 1 or 2 weeks. And in that we show that CB2 also can protect in a cultured human CB1 LCA model, retinal organoids. So I do not have the time to show these, but so CB2 can protect the retinitis pigmentosa retina, and also very likely the LCA retina. At least that is shown in our most recent data. So the very first phenotype that we observe in CB1 knockout retinal organoids is very similar. So in the patient retinal organoids, the phenotype is very similar. Because we see just that rows of photoreceptor cells, they let loose from so nuclei from the photoreceptor layer, they let loose and they are misplaced into the cell medium when we look into cultured retinal organoids. So the phenotype, what we observe in the CB1 knockout mice, is very similar as that we observe, for example, in CB1 patient organoids, but also as what we observed, so patient retinitis pigmentosa models, but also when we look into CB1 knockout human retinal organoids. So these phenotypes are very much the same in mouse and in human. In 2019, we did publish a paper in Stem Cell Reports, and there we showed that iPS-derived retinas, that they recapitulate the fetal CB1, CB2 complex formation. And what we observed in these retinitis pigmentosa retinal organoids was that photoreceptor nuclei, they were misplaced, they were on the wrong side of the so-called outer limiting membrane. So they were not anymore, nuclei were not anymore in the outer nuclear layer, so the photoreceptor outer nuclear layer, they were misplaced. So we observed that in mice, and we observed that in 2019 in our patient CB1 derived, patient-derived retinal organoids. Well, and we now did further studies on these. So the patients that we used, there were, for example, a cysteine 948 tyrosine. They also were patients with a methionine 1041 threonine, with a glutamine at 995 mutated, and the tyrosine 631 cysteine. So these were common mutations found in the human population. So we had these patient lines generated, and we used CRISPR-Cas to make some proper controls. So some so-called isogenic controls. That means that the genetic background of the patient and of the control, they are the same. These are isogenic controls. We had these isogenic controls cultured together with the patient retinal organoids, and these structures looked very nice. We had differentiation day 209, 210, so these were cultured for 7 months, for example. Sometimes we cultured for 8 months, and these retinal organoids really look beautiful. But yeah, so as told, the patient retinal organoids, they show a retinal phenotype. The number of photoreceptor nuclei observed in the patient organoids, the number of nuclei was decreased. Also, the outer nuclear layer thickness was decreased, and we at differentiation day 180 saw an increase in number of photoreceptor nuclei above the outer limiting membrane. So that was very nice. So we did have a proper control and proper patient's retinal organoids. Then we looked again and compared these isogenic organoids with the patient organoids, and to our surprise was we found that the levels of the CRB1 protein was strongly reduced in the patient lines. So we had 3 different patient lines. We called them the patient lines 116, 117, and 128. So these patient lines, they had about 5-fold lower levels of the CRB1 protein. That was very surprising, because yeah, so that's normally not found. Then there's just a change in one of the building stones. So it's not a knockout. It's just that there are variant amino acids, variant building stones in the protein, and so that was very surprising, but an interesting finding. So we further analyzed that. We really did these experiments several times, and we quantified this, and there's about 5-fold reduced levels of the CRB1 protein. We used different antibodies. We did not believe that, and yeah, but we confirmed that with different antibodies, and yeah, a real reduction in the levels of variant CRB1. Then we use a technique that is called single-cell RNA-seq, where we could put tags to the cells and tags to the transcripts in the cells, and we could separate the cells from each other. So we could have separated molecular cells, separated rod photoreceptors, separated cone photoreceptor cells, and also other separated retinal cell types. We did many of these, a lot of bioinformatical studies on these, could have this nice separation into these different cell types. That worked out very nice. Some of the cells we also infected with a fluorescent marker, and these studies showed us that we also infected molecular cells. When infected with AVs, we could infect molecular cells, we could infect rod photoreceptor cells, and we could infect cone photoreceptor cells. So, and thereafter we did AV. First we further analyzed all of these cell types. But what we at a certain time did was we used a phi factors to express the CB1. I just told that these give substantial levels of CB1 expression in the mouse, and we also showed that we have CB1 expression in these CB1 retinal organoids, and we I forgot to tell, that's just when I do things by head. We also look for CB1B transcription level. Well, to our surprise, there was absolutely no CB1B protein expressed in these very early retinal organoids, but there was already a phenotype. So, the CB1A form, or the full-length CB1 protein, has a very important function in myeloglial cells and photoreceptor cells. At these very early stages, there's no expression of the CB1B at all. So we found that in our single-cell RNA-seq data, but we also look into that by qPCR, quantitated PCR. So the important players are the full-length CB1 proteins. The CB1 full-length CB1 gene products. We looked and just if there are any differences in levels of the CB1 in the isogenic control compared to the patient, and just we did not see any differences in transcript levels of in the myeloglial cells or in photoreceptors, not in the cones, not in the rods. We also did not see any differences in transcription levels for the CB2, and also not in other genes that play a role in the function of the CB1 complex. We then looked into any changes into the gene expression. First, we did a comparison of the control with the patient material, and we found that biochemically there's something changed in the endolysosomal system. The endolysosomal system is of high importance to bring the CB1 protein, for example, to the outer limiting membrane, and also the recycling of this protein. That was of high interest, that we found these changes in the endolysosomal system. When we then looked into the retinal organoids, the CB1 retinitis pigmentosa retinal organoids that were infected with the CB1 or infected with the CB2, we saw that the number of photoreceptor nuclei significantly improved when treated with the AV-CB1 or CB2. And also we observed that this number of photoreceptor nuclei above the outer limiting membrane also significantly improved when analyzed at differentiation day 180. Also, when we looked into at later time points, differentiation day 210, we saw significant improvement of the number of photoreceptor nuclei in a row, and also in the outer nuclear layer thickness. So CB1 and CB2 both protect against retinal degeneration in cultured patient CB1 retinal organoids. We then also did gene profiling studies on myeloglial cells and photoreceptor cells treated with the And we observed that the changes in endolysosomal, so the endolysosomal aberrations observed in the patient was protected. Yeah? So we saw that just the gene expression levels, which were abnormal in the patient, just in the treated patient, this CB1 or treated with CB2, we saw that the gene expression profiles were very similar to the isogenic material. So that was really exciting. So that was the case for as well the CB1 as CB2. And we did these studies on multiple patient derived, multiple independent patient material. So we observed that for patient number 128, but we also observed that for patient number 116. For both, we had isogenic controls. So, in summary, and the story that I now told is for the ones interested. This has been published recently in the journal Stem Cell Reports, and the first author is Nanda Bohm. And what we've shown is proof of concept for AAV-CB2 in 3 different CB1 mouse retina. Those were the old data. So in 3 different mouse models, we showed that CB2 can protect against retinal degeneration caused by mutations in the CB1. Now, we have also proof of concept that in human retinitis pigmentosa model, the CB2 can rescue, can protect against retinal degeneration. And what I did not tell about today is that the CB2, but not the CB1 factor, can also protect the human LCA retinal organoid models. So we have strong proof now that the CB2 is able to protect against loss of function due to mutations in the CB1. In the past years, already from 2017, my colleague Camille Bohn, I was also co-author on several of these manuscripts. In 2017, they reported the first natural history study on patients with mutations in the CB1. There were publications in 2017 and 2021, 2 other papers. Those were retrospective studies. And in 2021, and there's also an upcoming paper by Jessica Carunto, and these papers, they describe a prospective study on CB1 patients. And I hope that Jessica is here, that Jessica can inform us about the clinical natural history studies. Yeah, Jessica is there.
Yep, thank you, Jan, for the introduction. So first, I would like to thank the organization for inviting me for a very short talk about our clinical studies, and for the participants for listening and participating in this meeting. So I'll start with this very short introduction. I'm Jessica. I'm a PhD candidate from the Netherlands. I have an MD, and we've been working on CRB1, retinitis pigmentosa, macular dystrophy, and LCA for a few years now. What we're doing is we're looking into natural history studies. What does that mean? That means that we want to investigate the disease course. So what happens in the retina? What can we see on our scans? How does that progress further over time? What happens to visual acuity? What happens to the visual field? And we're mapping all of that out. And why is that important? Because CRB1 retinal degeneration is just like any other retinal degeneration. That is true, but at the same time, it all depends on which genetic mutation you have. So the disease course can be completely different. The time points when you reach, for example, legal blindness can be completely different from another genetic mutation. So that's why we're doing these natural disease course studies. Or furthermore, also interested in, can we investigate the clinical variability? Can we investigate which tests we can use if we want to start to the clinical trials? So, for example, are the tests that you are doing, the visual acuity test, are those relevant to actually measure the retinal degeneration, or should we be using other tests? So that's very short what we've been investigating. And like Jan also said, we've published a few studies already. The first one is done by my colleagues. I only started in 2021, and we've published that. And if you look under the name Camille Bone, then you can find most of those studies. So the first paper that we've published was on a Belgian cohort. We've also done this for a Dutch cohort. And although it may not be completely the same across the globe, it will give some sort of guideline as to what you can expect. And so these first studies were all retrospectively, which means that we just look into the patient files. We look from the first time they went to our clinic, we see when they came back and what happened then. So it's all looking back into the past. And already by doing that, we already included about 40 families, 40 patients. We found that, for example, patients with any type of CRB1 retinal dystrophy can get more insight into their visual prognosis. For example, generally we found that low vision is found about 18 years, severe visual impairment around 32 years, and legal blindness before 44 years. But again, this is all on a population level. This is not what you can expect as an individual. It may differ, it may vary over time, and it may vary per person. So this is very general speaking. And then, of course, we're also interested in if you want to translate the research from Dr. Jan Wijnholt, if you want to translate that to the clinic, when should we do that? What is the best time to do that? Do you want to treat people who still have a great visual acuity, there's nothing wrong, who just got the diagnosis, or should you treat people who are already severely visually impaired, and should we treat those first? What is the best time to treat, and will it still be useful if you treat them with gene therapy? So we found that, for example, looking into the visual prognosis, we suggest with people with RP, you should treat them before 30 years. And again, this is a very broad definition. It is not applicable to everyone, and it really depends per person. And this is just a guideline, because this is about what we see when we look at all the retinal structures, we look at the visual acuity, we look at the visual fields. This is somewhat what you can expect. Now, for example, if you have Leber's congenital amaurosis, which is another part of a retinal degeneration, we suggest starting the therapy before 10 years. And it's all suggestion. And then later, so this is all done based on studies we've done by looking into patient files, looking into the past. And there's a lot of missing data as well, because you're looking at in the past. So what we did is we started a prospective trial, which means that we start following patients. We see them at regular time intervals. We do more extensive examinations. We do more functional examinations as well. And we're looking into which measurements are the best to use if we want to start clinical trials. And we found, for example, and these are also published by Camille Bone's group, we found that, for example, visual acuity and visual field, they're relatively stable over the course of 4 years. So if you want to use it as a clinical endpoint to measure anything, it's not very useful. But for example, microperimetry, which is a test which measures the sensitivity of the retina, specifically of the central part, so the macula, that is a far more sensitive measurement, and we can already see the decrease in sensitivity, even when the patient does not already notice that. So that is one of our conclusions. So we suggest using the microperimetry, so the macular sensitivity, as one of the clinical outcome markers. And we've made suggestions for the therapeutic window. Now, there is another paper which is not done by our group, but is from the Moorfield Eye Hospital. And they've also studied a lot of CRB1 patients, but again, by looking into the past. So they've included about 100 patients. And they also, most of their findings are similar as to ours, but they suggest, based on their patient populations, that the people with Leber's congenital amaurosis can be already treated even after 10 years, but before the age of 20. And again, also for them, it's also a very big guideline. It doesn't say that you're not eligible for treatment anymore if you're 21. But it's very individual. There's a lot of individual variability there. And if you have any questions regarding the clinical studies, you can always contact me. And I'll be happy to answer any questions if you have those.
All right, I think that we're going to shift. So hi everyone, my name is Jason Menzo. I'm the CEO here at the Foundation Fighting Blindness, and I want to thank all of you for spending time today here on a Saturday. We've got time zones really across the globe covered today. We've got folks as far west as Hawaii and as far east as Western Europe, and so it's really a special opportunity we have to have this terrific group of speakers. Jessica, Dr. Winholtz, Angela, it's terrific to have each of you on the call here today. Jan, in particular, I want to thank you. You know, when I first joined the foundation and first started learning about CRB1, there was one name that was repeated over and over again from our constituents, and that was your name as one of the pioneers, really diving into CRB1 research at the very, very early days. It's terrific to see how much progress is happening in this field. And so I want to thank you personally for the work that you did and continue to do to push the envelope and push forward, you know, potential treatments for this specific condition. It is, you know, we're running a little short on time. So what we're going to do is, rather than have a full-blown Q&A, I'm going to ask, first of all, I've captured all the questions that have been chatted in so far. And so we will follow up with individual answers to those questions that have been chatted in so far. Additionally, anyone who has a question that didn't pose it yet, I'm going to give you an email address right now that you can send any question that you have related to this webinar, anything related to the work we do at the Foundation Fighting Blindness, and we will get back to you individually. That email address is info@fightingblindness.org. And again, any question you have, whether it be about CRB1 or this webinar today, or really any other work that we do here at the Foundation, we have a team that will follow up with you individually on any question, including those that have been sent in today. Before we have our closing remarks, I do want to pose one question because we heard some terrific work coming out of Dr. Winholtz's lab. Really exciting work, Jessica, that you're doing with regards to the natural history study for CRB1 and looking at multiple phenotypes. That's exciting. But you know, at the very beginning, Tabby posed, referenced the fact that we have this new person on our staff in this new position in Dr. Angela Bowman, that part of her responsibilities are to assess what's happening in the landscape across a small portfolio of different genes, CRB1 being one of the 3 genes that she's focused on. And Angela, before we close out the call, I think it'd be really great for our attendees today to hear from your perspective some of the highlights that you see in the field across the board that people should be aware of, and perhaps even optimistic about the progress that's happening in this field.
Sure, I'd be happy to. Thank you, Jason. This is Angela Bowman speaking. Thank you so much, Jan and Jessica, for speaking. Your research is just the kind of research we need for the CRB1 community. Thank you, Jan, for talking about the long history you have with CRB1 and all the detailed work you've done to understand CRB1 and CRB2 and how they're connected and how they can compensate for each other. And so I think the combination of work that you've done in the mouse and gene therapy in the mouse as proof of concept, and now applying these kind of newer technologies like retinal organoids and what we can learn from human mini organoids in the dish and how that will advance retinal therapies for CRB1, I think is just so exciting. And as I talked about in my introduction, Dr. Winhold's former student, Peter Quinn, is really applying some of these most advanced cutting edge technologies with prime editing. And I know that we like to have multiple shots on goal. So a gene therapy is great, but maybe prime editing will also be great for other people or for other reasons. So I'm so excited that the field is expanding and that we have a new pig model coming online, hopefully. And just all these tools in our toolbox will really help us, I think, move the field forward.
That's great. Thank you so much, Angela. And I do have just a couple final remarks here this afternoon before, I should say this morning or afternoon, depending on where in the world you are joining us today. First of all, this entire webcast is being recorded. And so if there's anything that you want to revisit, something you wanted to re-listen to get a little bit more detail, certainly you'll have that option. We're going to follow up with an email with some additional information to all attendees and everyone who registered. So that's one thing. I do want to remind everyone, like I said a minute ago, any question that you didn't pose or that we didn't get to, or another question that you think of, you can contact us anytime at info@fightingblindness.org. And then I just wanted to say that the Foundation Fighting Blindness really continues to be a global leader, not just as it relates to broad IRD research in general, but really, I want to talk specifically about the impact that we've had on CRB1 specifically. We've invested millions of dollars specifically towards CRB1 research, and we continue to look for new and innovative ways for us to impact the field as it relates to CRB1 specifically, whether that be additional animal models, as Angela referenced just a few minutes ago, or perhaps an intradisciplinary type of program project award, or perhaps even a translational research award, or maybe even an investment via our RD fund. So what's unique about the Foundation Fighting Blindness is that we've got so many different tools that we can leverage to bring whatever is needed specifically for a particular area of the inherited retinal disease landscape, to pull those levers to try to advance those specific areas that will benefit by using the tools that will benefit that area the most. And so with that in mind, and we heard it earlier today, and I want to make mention about how unique and special this opportunity is. Tabby, it is so, and it was a surprise. This is no tongue in cheek, no lip service. There was a surprise at the beginning of this call when Tabby said that Tabby and her company are willing to help raise money specifically for CRB1 research within the Foundation Fighting Blindness. Tabby mentioned, I wrote the number down, up to $100,000. So Tabby, thank you so much. And I want to share with the group here today how that would work. So with this very special, unique opportunity, anyone that wants to make a donation for the next 2 weeks to a specific page that the Foundation Fighting Blindness is set up, every dollar that is donated via this page will be restricted specifically for CRB1 research. This is a unique thing. We don't do this typically, but with this generous offer from Tabby, it only makes sense for us to try to take advantage of this opportunity to raise as much money as we possibly can specifically for CRB1. So how this works is that on your screen right now is a QR code, and you can either hold your phone up to that QR code and tap on it, and it'll take you directly to a specific page that will allow for you to make a donation that will be set aside specifically for CRB1 research. There's two ways you can get to that page. You can either click on the QR code, or you can directly type in www.fightingblindness.org/CRB1, or you can also go to donate.fightingblindness.org/CRB1, and that will take you directly to a page that has been set up specifically to earmark any money that's raised for the next 2 weeks, and that money will be set aside specifically for CRB1 research. And again, it doesn't matter the size. If it's $1, it's $1. It's fine. We want to be able to leverage as much as we can of Tabby's generous offer. And if you are outside the US, because I know this is a global conference call or a webinar that we're having right now, please contact Judy Taylor. She will be sending an email to all participants with this information, and she can also help facilitate a donation if there's anyone from outside the US who'd like to make a donation via this channel. So with that, I'm going to ask Jason Morris just to say a few words to close out the call. Jason is one of our board members and an officer at the Foundation Fighting Blindness, and has a personal connection not just to inherited retinal disease, but specifically to CRB1. And so Jason, I'll let you say a few words and then we'll close the call.
Thank you, Jason, and thanks to all the presenters today. Jan, it was particularly good to see you again. I've been to Jan's lab in the Netherlands, and can't tell you how fortunate we are to have him on the case of the CRB1 puzzle. So thanks to all. I hope you found the conversation interesting today. I am grateful to serve as a board director for the FFB. I am treasurer of the board of the FFB, and I can tell you our biggest responsibility is to protect the reputation of the Foundation Fighting Blindness. And part of the way we do that is through earning the trust of people who donate money to the FFB, and the rigor that is put forth by our scientific advisory board that I think it consists of over 60 people currently, is applied across every project that the FFB funds. And nothing is more important to us than making sure that the money is well spent and goes to things that will be successful or drive progress in the field. And that certainly will apply to the dollars raised today specific to CRB1. Thank you, Tabby, so much for your tremendous generosity and all of your family's support. Super exciting to hear that there's progress everywhere across the field, like Angela touched on, and certainly specific to what Jan and Jessica presented today. Very exciting to see things going on there. I mentioned during our November CRB1 forum that we will continue to do these a couple times a year. We'll plan to exchange ideas and just further discuss the roadmap for how we get from where we are today to that end goal that we're all looking for, where there are treatments and cures for CRB1 mutations and the broader spectrum of blinding diseases as well. Like you, I am interested in seeing research move faster. And I promise you, every day, I think about ways that we can help do that. Please be sure to act quickly on the CRB1 funding page. As Jason mentioned, it'll close on May 31st, and any dollar amount helps. Thank you all so much for taking time out of your Saturday, whatever time it is, and wherever you are. As Tabby mentioned so eloquently, whether we like it or not, and for FFB staff a little different, but whether we like it or not, we're in this fight together, and we are going to win. So stay positive. Let's keep moving forward, and together we can make a huge difference. Thank you.
Thank you, Jason. Thank you, everyone.